Right now, as you're listening to this, there are billions of microscopic, totally blind collisions happening inside your
Oh,
bloodstream.
absolutely. It's just constant chaos in there.
Right. Like, think about a chemical from the coffee you just drank, or maybe the ibuprofen you took for a headache. It's just floating in total darkness.
Yeah, bumping into thousands or, I mean, probably millions of your cells,
Exactly, just drifting along until it finds the exact microscopic lock it was designed to open.
which is pretty incredible when you think about it.
It is, because usually within like 20 or 30 minutes, you feel different. The headache fades or the brain fog lifts. But have you ever stopped to think about how utterly bizarre that actually is?
Most people definitely don't.
Right. I mean, how does a blind chemical know exactly where to go and exactly what to do? So today we are setting out on a mission to decode these bizarre biological handshakes that basically keep you alive.
It's a great topic.
We're doing a deep dive into a massive stack of pharmacological history, medical case studies, and biochemistry research, all to understand the locks and keys of our bodies.
And it really is a feat of biological engineering that we just take for granted every single day.
For sure. So where do we start?
Well, to understand those chemical handshakes, we have to introduce two foundational concepts first. These govern almost everything we'll discuss in this deep dive:
Okay, lay them on me.
the receptor and the ligand.
Got it.
So you can think of a receptor as a highly specific macromolecule. It's almost always a complex protein.
And where are these proteins?
Usually they're embedded right there in the outer membrane of your cell,
Mm-hmm.
though sometimes they sit inside the cytoplasm or even the nucleus.
Okay.
A receptor's entire job is to recognize a chemical signal, grab onto it, and then translate that binding event into a tangible change in the cell's behavior.
Right. And the chemical signal is the ligand.
Exactly. The chemical signal that does the binding, whether that's a naturally occurring hormone in your body, a neurotransmitter, or, a synthetic drug you just swallowed, that is called the ligand.
Okay, let's unpack this.
Yeah.
Because the idea of a physical protein sitting on the surface of a cell, just waiting for a specific chemical ligand to dock with it, that sounds so incredibly modern to me.
Yeah, it does sound like that.
Like, it sounds like something we must have figured out with an electron microscope, maybe 10 or 20 years ago. How did we actually discover these invisible locks in the first place?
See, that is actually one of the greatest ironies in the history of medicine.
Really?
Yeah, the concept of the receptor is quite old. Way before electron microscopes. We have to go all the way back to around 1900.
Oh, wow. That early.
Yep. To a German chemist named Paul Ehrlich. He was looking at how certain synthetic dyes would stain some tissues in a petri dish, but then they'd completely ignore others.
Mm-hmm.
He was also studying early treatments for syphilis and sleeping sickness. He was trying to figure out why certain arsenic-based compounds would kill a microscopic parasite but leave human cells completely unharmed.
Because it's targeting something specific.
Right. So Ehrlich coined a Latin phrase, corpora non agunt nisi fixata, which roughly translates to agents do not act unless bound.
Which makes intuitive sense, right? A drug can't do anything to a cell if it just floats past it in the bloodstream.
Exactly. It has to actually connect. So he proposed that cells had these specific side chains sticking out of them, and that a chemical had to physically dock onto one of those chains to have any effect.
Okay, so that's the basic theory.
Yeah. And right around that same time, slightly earlier, actually a British physiologist named John Newport Langley was running these wild experiments at Cambridge.
What kind of experiments?
Well, he was testing nicotine and the arrow poison curare on the skeletal muscles of frogs and cats.
Wait, curare, like the poison used on blow darts by South American hunters, the stuff that paralyzes animals?
The very same. The active compound is tubocurarine.
Wow, okay.
So in 1905, Langley made this profound, really groundbreaking realization. He knew applying nicotine caused the frog's muscle to contract.
Right.
And he knew applying curare blocked that contraction, effectively paralyzing it.
Because it's a poison?
Exactly. But his key insight was that the curare didn't paralyze the muscle by destroying the nerve. The nerve itself was totally fine.
Wait, really?
Yeah. And it didn't damage the muscle fiber either, because if you bypassed the nerve entirely and just stimulated the muscle directly with electricity, the muscle would still contract perfectly.
Oh, that's fascinating. So the poison was stopping something exactly in between the nerve ending and the muscle?
Precisely. It intercepted the signal right at the border. It blocked the specific site of interaction on the muscle surface.
The handshake.
Right. It blocked the handshake. And Langley called this the receptive substance, which is, the direct linguistic ancestor of our word receptor.
That is so cool.
It is. Then, in the 1920s and 30s, a researcher named A.J. Clark came along and formalized all of this mathematically.
Okay, bringing in the math.
He created what's known as occupancy theory, basically proposing that the magnitude of a drug's effect is directly proportional to the fraction of these receptors it occupies.
So the more locks you fill, the bigger the effect?
Exactly. And this gave pharmacology its very first quantitative dose-response curves. You could chart out exactly how much drug equaled how much biological effect.
Okay, so they have the math figured out. They have the dose-response curves. They have this theory of a receptive substance from the frog experiments. And tell me if I'm understanding this right from the research, it seems like there is this massive gap in the timeline here.
There absolutely is.
Right. From 1900 all the way until the 1970s, no one had ever actually seen one of these receptors.
You read that completely right. For about 70 years, the receptor was essentially a highly useful mathematical fiction.
A ficky.
Yeah, it was a hypothetical entity inferred purely from those dose-response curves. Scientists were designing drugs for a target they couldn't physically prove was even there.
That is wild.
It wasn't until the 1970s that scientists finally had the technology to isolate, purify, and sequence these physical proteins in a lab.
So studying receptors for those 70 years was basically like studying a black hole.
Oh, that's an interesting way to look at it.
Like you couldn't actually see the receptor itself, but you knew it had to exist because of the gravitational pull it had on your data, like the frog's muscle reacting or not reacting.
That is a brilliant way to put it, yeah.
Yeah.
You can't see the black hole, but you can see the light from the stars bending around it. They couldn't see the receptor, but they could see the dose-response curve bending around its presence.
That makes perfect sense.
But here's the thing. Once scientists finally proved these invisible black holes existed as physical proteins, they realized that a drug binding to them isn't just a simple on-off switch.
It's not.
No, the interaction is far more nuanced. It's controlled by two completely independent dials.
Okay, two independent dials. Walk us through how those work.
Sure. So the first dial is called affinity. Affinity is a measure of how tightly a drug sticks to the receptor once it bumps into it.
Okay.
We usually measure this using something called the dissociation constant, or KD. A low KD means the drug.
Whoa. Let's pause right there. Dissociation constant sounds like some pretty heavy biochemistry.
Fair enough, yeah.
For those of us without a lab coat, you're basically just saying this is the measurement of the drug's stickiness.
Yes, exactly. Stickiness is a great word for it.
Hmm.
It measures how well the key fits into the lock and, crucially, how hard it is to pull it back out.
Got it.
If a drug has high affinity, it means it's very sticky. It takes very little of the drug floating in your blood to occupy half of your cell's receptors.
And low affinity.
Low affinity means it just doesn't stick very well, so you need a massive dose to see an effect. But affinity is only the first dial. The second dial is efficacy.
And what does efficacy measure?
Efficacy measures how much of a biological response the drug actually produces once it's stuck there.
Okay.
Like, does it turn the cell's machinery on just a little bit, or does it crank it all the way up?
Oh, I see.
Because these two dials, the stickiness and the biological response are completely independent, a ligand can score high or low on each one. And this gives us four broad categories of drugs.
Okay, what's the first one?
First, you have your full agonists. These have high efficacy. They bind and they produce the maximal system response.
Like what?
Well, epinephrine hitting your beta-adrenergic receptors and spiking your heart rate during a fight or flight moment. That's a perfect example.
Okay, so if we imagine a volume dial, a full agonist turns the volume all the way up to 10.
Right. Then you have the second category, partial agonists.
Yeah.
These bind to the exact same site, but they have lower efficacy.
So they don't turn it to 10.
Exactly. Even if they occupy 100% of the available receptors on the cell, they cannot produce the full maximal response. They hit a biological ceiling.
Interesting.
Buprenorphine, which binds to the mu-opioid receptor in the brain, is a partial agonist.
Wait, so if a partial agonist hits a ceiling, does taking more of the drug push past the ceiling? Or is it physically capped at a, say, 5 out of 10 no matter what?
It is physically capped no matter how much you take. You can take 10 times the normal dose, and the volume will not go past a 5.
Wow.
And that ceiling effect is precisely why buprenorphine is vastly safer in an overdose situation than a full agonist like, say, heroin or morphine. It simply cannot trigger enough respiratory depression to stop your breathing.
That makes total sense. What's the third category?
The third is the inverse agonist, and this one is really wild.
Lay it on me.
An inverse agonist binds to a receptor and actively reduces the receptor's baseline signaling below its resting level.
Below its resting level.
Yeah. It proves that many receptors in your body aren't just sitting totally dormant. They're actually constantly humming with a low level of measurable activity, even when nothing is bound to them.
Oh, like a quiet static on a radio, and the inverse agonist actually reaches over and turns the radio off completely.
That is a perfect analogy, yeah. And finally, the fourth category, the antagonist.
Sounds villainous.
Well, it's very useful, actually. An antagonist binds with real affinity, meaning it sticks to the receptor well, but it has absolutely zero efficacy.
Zero, so it just sits there.
It just sits there. It occupies the site and blocks the full agonists from getting in, but it doesn't trigger any biological response itself.
Wait, so a highly effective drug isn't necessarily one with high affinity. That feels a bit backwards to me. You'd think the tightest binding drug, the stickiest one, is the one doing the most work inside the cell.
It's completely counterintuitive, right?
Yeah.
But what's fascinating here is that high affinity does not equal high action. Affinity only measures how well a drug sticks. It says absolutely nothing about what happens next. And this distinction between affinity and efficacy is quite literally the only reason we are able to fight back against the opioid crisis.
You mean with fentanyl and naloxone?
Yes, exactly.
Let's break that down, because I know a lot of people have heard of Narcan, but they might not know how it actually works mechanically.
Right. So fentanyl is deadly because it has ultra-high potency as a full agonist at the new opioid receptor. It's roughly 50 to 100 times more potent by weight than morphine.
Which is terrifying.
It is. A dose in the low milligram or even microgram range can easily be lethal. It binds, and its efficacy is so high that it cranks that volume dial way past 10, which completely shuts down the brain's respiratory drive.
The person just stops breathing.
Right. But the rescue drug, naloxone brand name Narcan, saves lives purely through the math of affinity and efficacy.
How exactly does it do that?
Naloxone has an incredibly high affinity for that exact same opioid receptor. Its affinity is so much higher than fentanyl's that it can physically rip the fentanyl right off the receptor and take its place.
Wow, it just rips it off.
It aggressively outcompetes it. And this is the crucial part, naloxone is an antagonist. It has zero efficacy.
Oh, I see.
It doesn't produce any opioid effect itself, so it kicks the fentanyl out, stops the signal that's suppressing the breathing, and reverses the overdose within minutes.
So naloxone is like a jerk taking the last parking spot that's the high affinity, the stickiness, but then just sitting there with the engine off, completely blocking the dangerous fentanyl race car from parking, which is the zero efficacy.
I love that. Yes, it aggressively steals the parking spot and refuses to turn the engine on. High affinity plus zero efficacy is exactly what makes a life-saving antagonist.
That's amazing.
It is. But understanding how these drugs fit into these molecular parking spots forces us to confront an uncomfortable truth about how we historically visualize this whole process.
Uncomfortable how?
Well, for a long time, science pictured this interaction as a rigid key fitting perfectly into a rigid lock. Have you ever seen that diagram?
Right, the lock and key. Yeah, I remember seeing that exact diagram in my 10th grade biology textbook. A perfectly rigid, jagged piece sliding into a perfectly rigid lock. Are you saying that's wrong?
It is entirely wrong. Though, to be fair, it was a very useful stepping stone.
Okay, so where did it come from?
A scientist named Emil Fischer proposed that lock and key model way back in 1894 for enzymes, and we just sort of ported it over to receptors. It implied this rigid, unmoving shape.
Which isn't true.
No.
Mm-hmm.
In 1958, a scientist named Daniel Koshland updated it slightly with the induced fit model. He proposed that the receptor changes shape slightly in response to the ligand binding.
Like what, sort of like a hand molding a flexible glove around it?
Exactly like that. Better, but still not the whole picture.
Okay, here's where it gets really interesting. Because didn't a relatively recent Nobel Prize completely rewrite how we view this?
It did. In 2012, Brian Kobilka and Robert Lefkowitz won the Nobel Prize in Chemistry for their work on G-protein-coupled receptors.
And what did they find?
Using these advanced imaging techniques like crystallography and cryo-electron microscopy, they proved the conformational ensemble model. The what now? The conformational ensemble. Basically, they showed that receptors are not rigid locks, and they aren't even passive gloves. Even when they're completely empty, receptors are constantly jittering, vibrating, and flickering between multiple different 3D shapes.
Just on their own.
Just on their own. A ligand doesn't force a new shape into existence. It simply binds and shifts the probability of which shape the receptor will spend the most time in.
Okay, so instead of a rigid lock, it's more like a dancer constantly shifting between different styles. salsa, tango, waltz.
Yeah.
And the drug doesn't force a specific pose, it just acts like the DJ. If the drug plays a Latin beat, the receptor spends 90% of its time doing the salsa, which opens door A inside the cell. If a different drug plays a waltz, the receptor changes its rhythm, which opens door B.
If we connect this to the bigger picture, that DJ and dance floor analogy is exactly why the field of biased agonism exists today.
Biased agonism.
Yeah. Under the old rigid lock and key model, if two different drugs bound to the exact same receptor, they should theoretically trigger the exact same effect.
Because the lock only opens one way.
Right. But because receptors are these dynamic, jittery ensembles, two different drugs can hit the exact same receptor, favor slightly different shapes, play a different beat, as you said, and steer the cell down completely different biological pathways.
Which is wild. Two keys go into the same lock, but they open completely different doors inside the house.
Exactly. And clinically, this is huge.
How so?
Well, for instance, drug developers are currently engineering experimental opioids that bind to the mu-opioid receptor. But they favor the downstream pathway that provides pain relief and deliberately avoid triggering the pathway that causes respiratory depression.
So they're trying to separate the pain relief from the lethal side effects all at the exact same receptor?
Exactly.
But wait, if these receptors are constantly dancing and jittering and they belong to these massive overlapping families of proteins, how on earth do chemists design a drug that hits just the right one?
It's not easy.
It seems like you'd accidentally trigger a dozen different pathways in the body if the drug bumped into the wrong dancer.
That is the greatest challenge in pharmacology. And it brings us to the concept of selectivity.
Okay.
Selectivity is the holy grail of drug design. Because receptor families contain multiple subtypes with binding pockets that are very similar, but not perfectly identical, chemists try to tune a molecule to prefer just one subtype.
Do we have examples of that working?
Oh, definitely. The textbook success story here is James Black, a Scottish pharmacologist who won the Nobel Prize in 1988 for rational drug design.
What did he design?
In the early 1960s, he engineered propranolol. It was the first clinically useful beta blocker to protect the hearts of angina patients.
Okay.
But propranolol was non-selective. It blocked beta-1 receptors in the heart, which lowered blood pressure, but it also blocked beta-2 receptors in the lungs.
Which I'm guessing is bad.
Yes, blocking those beta-2 receptors could trigger dangerous bronchospasms, making it very risky for asthma patients.
Ah, I see.
So later, chemists used Black's exact logic to engineer metoprolol, which is highly selective for just the beta-1 receptor in the heart, completely sparing the lungs.
That's brilliant.
Black also used this logic to invent Semidyne, targeting the H2 histamine receptors in the stomach to cure ulcers, while completely ignoring the H1 histamine receptors that cause seasonal allergies.
So selectivity is the ultimate goal. You find the exact receptor subtype you want, you target it, you avoid the others, and everyone goes home happy. More selective is always better, right?
Well, this raises an important question, and it's a massive misconception in medicine. The idea that more selective automatically means safer?
It doesn't.
It doesn't. Selectivity just redistributes your risk.
How so?
Let's look at the painkiller Vioxx, which came out around 1999. Older anti-inflammatory drugs, like standard ibuprofen, inhibit two enzymes, KOX1 and KOX2.
Okay.
But hitting KOX1 causes severe gastrointestinal bleeding in some patients because KOX1 protects the stomach lining. So Merck designed Vioxx to selectively inhibit only KOX2, which drives inflammation.
Sounds like a good plan.
And it worked perfectly. It stopped the stomach bleeding. But that hyperselectivity shifted the balance of chemicals called prostaglandins in the body in a way no one fully anticipated.
Uh-oh.
By leaving KOX1 alone but totally blocking KOX2, the drug inadvertently promoted blood clotting and constricted blood vessels. By the time it was pulled from the market in 2004, it had been linked to tens of thousands of excess heart attacks and strokes.
Oh my God. So by focusing purely on selectivity, we essentially played molecular whack-a-mole.
Pretty much.
We successfully whacked the stomach bleeding mole, but because everything in the body is connected, we accidentally popped up a massive heart attack mole.
That is exactly what happened. You cannot tweak one isolated variable in a complex biological system and expect the rest of the system to just ignore it.
Wow.
We saw a very similar selectivity failure in 1997 with the diet drug combination fen-phen. It was pulled from the market because one of the compounds had an off-target affinity for a very specific serotonin receptor subtype, the 5-HT2B receptor.
Now, when I think of serotonin, I think of mood, or in the case of a diet drug, appetite suppression in the brain. Why was hitting this specific 5-HT2B receptor so dangerous?
Because that specific receptor subtype is also heavily expressed on the physical valves of your heart.
Oh no.
When the drug constantly stimulated those serotonin receptors on the heart valves, it triggered a biological cascade that caused the cells to rapidly divide and grow. It led to a thick, fibrotic buildup of tissue on the valves, making it impossible for them to close properly.
That sounds horrific.
It was, and nobody had screened for it because the drug was designed for the brain. They didn't realize the drug was also binding to the heart and causing permanent structural damage.
Now, when we talk about terrible drug disasters and the importance of getting the molecule exactly right, the one everyone learns about in school is thalidomide.
Right, of course.
The story is always told as this tragic failure of stereochemistry. The drug had a left-handed version and a right-handed version, exact mirror images of each other. The textbook always says one was a safe sedative for pregnant women and the other mirror image caused catastrophic birth defects.
Yep, that's the story.
And the moral of the story is always, if only the chemists had separated the two, the tragedy would never have happened. But based on the research we pulled, is that actually true?
It is a myth that absolutely refuses to die.
Really?
It is true that thalidomide exists as two mirror images, or enantiomers, but the genuinely surprising correction is that thalidomide's two enantiomers physically interconvert inside the human body.
Wait, they flip back and forth?
Yes. Even if you painstakingly purified the drug in a lab and gave a patient a pill containing only the safe mirror image, the moment it entered their bloodstream, their own liver enzymes and metabolism would start flipping a portion of it back into the dangerous teratogenic form.
Wow. So simply separating them in the factory wouldn't have saved anyone at all?
No, it wouldn't have. The body would undo all that hard work in a matter of hours. And what makes the thalidomide story even more sobering is that the actual receptor it binds to a protein called cereblin, which disrupts limb development signaling in the fetus, wasn't even discovered until 2010.
2010.
Half a century after the tragedy, molecular biology finally caught up to what the drug was actually doing on a cellular level.
It just shows how incredibly complex this all is. And a drug isn't always just acting as an agonist or an antagonist in a vacuum either, right? It depends entirely on where it is in the body.
Exactly. The cellular neighborhood matters just as much as the drug itself.
Like what?
Take tamoxifen, which is widely used to treat breast cancer. It's a selective estrogen receptor modulator, or SERM. In breast tissue, tamoxifen acts as an antagonist. It binds to the estrogen receptor and blocks it, preventing estrogen from fueling the growth of the tumor.
Okay, that makes sense.
But why does it block it? Because in breast tissue, the drug recruits proteins called co-repressors, which shut the system down. But if you look at the bone or the uterine lining, tamoxifen acts as an agonist, activating the exact same estrogen receptors.
How can it activate the exact same receptor or it just blocks somewhere else?
Because the bone cells contain different background proteins called co-activators. The receptor folds slightly differently around the tamoxifen, recruits these co-activators, and suddenly the drug is turning the cell on instead of off.
Oh, wow.
This protects the patient against osteoporosis, but it slightly increases the risk of endometrial cancer. The drug's behavior completely changes depending on the proteins present in the cellular neighborhood it finds itself in.
So what does this all mean? We've covered 70 years of mathematical ghosts, affinity, efficacy, jittering dancers on a molecular dance floor, and the very real dangers of playing whack-a-mole with selectivity.
We've covered a lot.
But this isn't just abstract science for researchers and lab coats. This is the exact reason you can walk into a pharmacy and buy a non-drowsy decongestant.
Absolutely.
The chemist specifically engineered that drug to target peripheral receptors in your nose rather than the central receptors in your brain that would put you to sleep. It is the reason a patient is prescribed a specific cardioselective beta blocker that lowers their blood pressure without triggering an asthma attack.
Right.
Understanding how these chemical keys fit into our cellular locks shapes almost every single medical decision made today.
It really does. But if you step back and look at the trajectory of everything we've discussed in this deep dive, it leaves you with a rather profound question about the future.
Ooh, what's that?
If receptors aren't rigid locks, but rather dynamic, jittery ensembles that can be steered down different pathways, and if selectivity is a double-edged sword where trying to hit just one specific target often causes unforeseen collateral damage, could the future of pharmacology move away from highly selective drugs entirely?
Wait, really? Away from them?
Think about it. Instead of manufacturing 10 different highly selective drugs for 10 different problems, could we one day engineer a single, highly complex master key drug.
A master key.
A drug that binds to a common shared receptor, but whose downstream pathway could be tweaked dynamically based on a patient's unique cellular environment.
A master key that adapts to the door it's opening, changing its function based on the room it's trying to enter. That is a wild thought to end on.
It's the next frontier.
Thank you so much for joining us on this deep dive. Keep asking questions, and we will see you next time.